welcome everyone to our 10th event for our translational neuroimaging educational program. For those who are joining for the very first time, we generally just do a quick introduction to the program itself, and then I'll be presenting our speaker for the day, which I'm really excited about. So in this program, we host a series of talks and presentations aiming to engage the neuroimaging and brain science community to enhance the translational application of computational neuroimaging techniques in promotion of human brain health. So you can find out more information using the QR code here, or check out the translational-neuro.org website to see more information about research that we post, information about us, as well as upcoming events. This educational program is nested within the Lifespan Working Group of the Enigma Consortium, and this is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. We hope to provide opportunities for you all to expand your skills, initiate collaborations, and facilitate networking in a welcoming learning environment. So myself and Dr. Ruyang Gi are the directors of this program, and we also have a core advisory committee that supports kind of the preparation and contact with various different researchers that have presented within this platform. On the website, you can also find a section where you can join our mailing list in order to receive updates of upcoming events. We also have a YouTube channel, and you can also follow us on X or Twitter, as well as Blue Sky. As I mentioned, we have a YouTube channel where all of the past presentations are posted. If you want to refer back to any of the talks that you may have missed, or especially for those who are finding out about this program for the very first time. So all the speakers' presentations are uploaded there. However, we encourage you to come in person because you'll get a chance to also engage with the speaker who's presenting, as well as ask questions, open up discussion at the end. We usually leave about 10 minutes for discussion. So have your questions prepared. And you can submit questions during the presentation in the chat, or you can wait till the end and unmute yourself and turn on your cameras to ask questions as well. And then again, the links are all on the website, but here you again have Twitter and Blue Sky, where you can follow us for upcoming events and details. And we're always happy to host speakers, especially those that are kind of at the early career stage, but also any topics related to translational neuroimaging, and we love to have talks that also have that kind of hands-on component. We have another scheduled event for March 27th. Dr. Yu Zhang, who's an associate professor of bioengineering and electrical and computer engineering at Lehigh University, will be giving a talk on data-driven neuroimaging biomarkers to empower precision mental health. So hope that many of you will also join for that event. But for today, I'm very excited to present Dr. Federico Raimondo, who is a team leader at the Institute of Neuroscience and Medicine 7, Brain and Behavior. His background is in computer science with a joint PhD in cognitive and clinical neuroscience. As part of the Applied Machine Learning Group, the Research and Infrastructural Software Engineering for Machine Learning, or RISE-ML, team focuses on the one hand, on applied machine learning research, exploring ways to improve neural-based machine learning models' performance, efficiency, and interpretability. And on the other hand, the team also works on infrastructural software development, creating robust frameworks, libraries, and platforms to facilitate applied machine learning research. We're very excited for your talk today, where you will be presenting Junifer and Julearn, from neuroimaging to machine learning models without expert-level coding skills. Thank you very much.
Hello. Oh, okay, so you hear me well? Yes, I guess. Okay, so thank you for that introduction. I found out when you invited me about this program. I think it's a great environment to promote all these kind of things that I'm working on. Mostly, our team focuses on doing this development of tools because we know that it's not easy to start a PhD and suddenly need to familiarize with all of these technical skills required to do this complex machine learning based and neuroimaging research. So I will start a bit blunt now. This is the first time I present these two tools at once, and I'm not quite sure of the background of the people on the other side and what they're expecting, but I will assume that there is some kind of knowledge of neuroimaging. So I will start with very basic things. So let's talk a bit about machine learning and what actually is. So the first part, when we are thinking about the experiments or classical statistics, we have data that we collect. For example, let's say that we want to see whether there is an intervention, and after, for example, not sleeping at all overnight, whether we are cognitively, let's say, a bit impaired in the morning. So we do a test in the evening, we don't sleep at night, and then we do a test in the morning, and then we compare the results, and then we do the same thing another day, but we actually sleep, and then we start modeling this. So basically, we have data and we have a hypothesis and the rules and what we expect that to happen. For example, we expect to fail the test only in the days that we haven't slept. So we perform our experiment, we collect our data, we set our hypothesis, and later on, we do an analysis, and this analysis will throw some results. Usually, we end up looking at p-values, and if the p-value is less than 0.05 or an arbitrary threshold that was set years ago, we decide and we conclude that our hypothesis is somehow true. So in this case, we are setting the rules, we are setting the assumptions on how the data is linked to the effect, and we get the results. Machine learning works in a slightly different way. So we have the data and we have the results. So basically, we have our tests, we have our subjects data, for example, some lifestyle or the amount of hours that the subject slept at night, and whatever variable we consider that might be linked to this effect of interest. And then we do an analysis, and what happens that machine learning actually comes with the rules for that to happen. So basically, the machine is learning how to give me the results based on the data I have, but we have to give the results. And this is called supervised learning, and this is what I'm talking when I'm going to talk about. So we're asking the computer to learn how to make an association between the data and the results without us giving the rules. So for the rest of the talk, when I'm going to talk about how we actually evaluate if a machine learning model works well. And let's start by data. We have imagine it's a table in which we have in our rows, we have the samples, we also call instances, examples. If it's, for example, human data, we can call them subjects, or samples in the same subject will be tested systematically. And on the columns, we have our variables, our features, our predictors, our any information of interest that we might think it might be linked to what we want to study. We call this usually the X, which is these variables are called X. It's a way that we note them when we write in machine learning terms. And then we have one of these becomes a special variable, which is the Y, and it says a lowercase y, which is the target or the variable that we want to predict. In the previous terms, how I showed in the previous slides, it was the results. So we have tabular data. This is what I'm going to focus on to explain machine learning. And this is our training data. This is where our machine is going to learn how to associate X with Y. And through the learning process, I'm not going to go into that, we're going to build what we call a machine learning model. So the thing that we need to understand about machine learning models is they are a bit like kids when you go to school. If you ask them if they learn, they will say yes. and the machine learning model is the same. They will learn rules to associate X with a Y, but the main question is what they actually learned about. They're very powerful. So one of the problems that we might have is that they actually memorize completely the data set. So you end up having rules that are too specific, and they're not general rules that will apply to any aspect. They just memorize that this subject identified by these variables will have this response. So it's not something that we would like, because the real goal of machine learning is to actually predict unseen data. If the machine learning model learned rules that applied general, means that apply in any aspect to the association between the variables in X and the variable in Y, they should apply also to the data that we have not seen yet. So we have our test data that we input, we try, we get the Y, and this Y is a predictive, it's a prediction of the label or that variable, given that we have the associated X variables. Now, but how do we know if our model actually worked? Because what we see here is that we try to predict, and this data should come from the future. So what we actually do is to simulate or take a peek at the future. So we take our data, but we leave some data aside. That is the data that would be in the future. We call this the train and the testing set. And then we train the model on the training data, which is a process. It's like a two-step process. The first one is called pre-process, is any data transformation steps that we might do. For example, a z-scoring, a centering, or complex stuff like doing a PCA to reduce the dimension. But any transformation on the data is done there. Then we learn. That's something magic that happens later. These are learning algorithms, and we give a model. And now we take this model. Here is the pre-processing, and we take this model and apply it to the testing data. That means that we transform the testing data in the same way that we train the training data, but without learning from it. So basically, we create this light purple vector, which is the predictive label, and then we compare, of course, if the label matches the actual labels that came with the testing data or not. So this is a way of measuring performance. Now, the problem is that this is not the actual performance of the model, because the model, to evaluate the performance, we need data that we have not seen yet from the future. This is an estimation of that performance, or the generalization error. And this is it. This is everything that we need to know to actually use machine learning. We need to understand that what we're doing is simulating the future to evaluate how well, or to estimate how well our machine learning model will work. And this is what we're going to report. The question is this going to be supported over time or not? If somebody else does this research again in the future and tries our model in their data, will this model work? And this is important, because we don't want to publish something that later on will be disqualified because it just didn't work. Let's take a couple of examples of neuroimaging based models to see how this could be framed. One that has taken a lot of attention lately is this brain age prediction. The principle is quite simple. We know that gray matter decreases with age, so the actual volume decreases, but to the extent of how is this decreasing, we are not quite sure. It could be many ways. So we have images of the brains from multiple people, and we also have the chronological age measured. For example, how many years old they were at the time of the scan. And then we put this into a learning algorithm that will give us a prediction of that age. And this is a plot that shows on the x-axis the true age of the subjects, on the y-axis the actual predicted age. Each one of these dots is a subject. It's one brain scan, an age pair. And what is shown here is that the correlation is 0.94. It's actually quite good. And this is called the mean absolute error, which shows on average how many years are we plus minus to the target. So, for example, on average, each person can be missed by 5 years, plus minus, which is actually quite good for this kind of models. Now, one thing that we can do with these models too is to compare the predicted age with the actual age. And for example, if we look at these two dots here, these green circles, we're going to see here that this is, for example, a 21, 22-year-old subject that was actually predicted at being 18. And this one is a 75, almost 80 years old subject that seems to be around 55 in terms of brain. So this could tell that, okay, it's quite a healthy brain, it looks even younger than it is. Now, if you're on these other dots, I have bad news for you. That means that your brain looks way older. Here it's not even 20, that looks like it has a 40-year-old brain, and this one is less than 50, and it has around 70 years old in the brain. And so far, this doesn't mean nothing. This is like just an error in the model. This happens because the model tries to learn general rules that needs to apply. But it has been shown that this could be used as biomarkers for aging. They can predict mortality. They can be useful for biomarkers of Alzheimer's disease and neurodegeneration, and also linked to Parkinson's and impairment. So these are quite, this is quite a research there that's happening based on machine learning. The first step was simple, just build a model that predicts the age given the brain scan. And this is just gray matter volume, parcellated gray matter volume. Another example, this is mine, actually one of the projects I'm working, and what we had is, on the one hand, we had a structural and functional brain image from the UK Biobank, so tens of thousands of brain scans, and the other way is a questionnaire about the overall quality of the sleep, but it's not super objective. It means that just questions placed that the subjects are answering related to whether they sleep well at night, yes or no, how many hours of sleep, whether they snore, whether they nap during the day. So the flow of this project is quite simple. So we take this self-reported sleep characteristics, and these are like answers that are a bit ambiguous. So we take the extremes and we create classes. So basically a binary variable, whether it's never, rarely I have never troubles falling asleep at night, or I usually have troubles falling asleep at night. So we take these subjects, and on the other side, we take the structural brain imaging and the functional brain imaging, and we do all sorts of measurements. We look at the cortical thickness, we look at the gray matter volume, and these are 3 measurements of a functional brain imaging that quantifies a bit about the local correlation, the global correlation, for example, the waves or of the Bolt signal in different parcellations, and we are both looking at cortical and subcortical information. So on the one hand, we have like proxies to the brain information, and on the other hand, we have like the self-reported sleep characteristics, and what we want to do is to see whether there is an association between them. So basically, if we have the brain, can we tell you whether you're sleeping well or not? This will be the first step. And if this happens, we can then go further and try to see why is this happening, like try to inspect the model. But the first step is always building the model. So we put this and we try several classifiers. We evaluate the performance using different metrics. And what we see is, this is like a bit of a spoiler, there is a preprint, but we could not predict, we could not find an association. But the result doesn't matter. What it matters is a bit about the workflow. And this is a typical machine learning workflow. We have data on one hand, genetic, behavioral, functional neuroimaging, structural neuroimaging, electrophysiology, any kind of data that can be acquired from a human in a cognitive neuroscience or genetic project. And then we process this kind of information, because if we know what this is, these are actually images, and these are a lot of pixels, voxels. So we want to try to extract meaningful information from them that we know that they represent actual physiological or biological processes. Like, for example, we can have this is about related potentials from EEG. This is cardiac information. We can have a tractography. This is functional connectivity. Any sort of information. And if you work with neuroimaging, you might know what I'm talking about. And on the other hand, we can also take from the same data, we can take the targets, for example, behavioral variables or genetic information, and then we can use machine learning to find associations between them. So this is typical workflow using machine learning and neuroimaging. Everything is fine, but now I'm trying I will try to scare you a bit. So if you work with neuroimaging, you will know that whatever comes out of the scanner, it can be used exactly like that. We need to do some sort of pre-processing. And if we're working with functional MRI, we have this fantastic tool, which is fMRI prep, which might look like we just take our data, we run a command, and then we have our data denoised, reoriented, everything in the same space and ready to be used. But if you actually work and see this slide, I'm not going to go explain it, we will see that there are many parameters that we can actually change. For example, do we filter, how we filter, how do we process, to which space we want to normalize. Do we do slice time correction or not? Do we do smoothing? So there are many things that can affect the results of the image, even though we haven't started even working with machine learning yet. And we've done an analysis, for example, and we compare how voxel-based morphometry, so basically extracting gray matter volume using parcellations, can be affected by the different tools that are used to compute. So if you know what I'm talking about, there are tools like ANTS, FSL, fMRI prep, and FSL. Then they use the CAT toolbox. And all of these, they have their own way of computing the voxel-based morphometry, or basically the gray matter volume, how much gray matter we have in each one of the parcellations of, for example, a Schaefer parcellation. And what we see here is this is the distribution of R values across the subjects for each one of the ROIs that we tested. And what we see, if you look, the ideal scenario will be something similar to what we have here, like a distribution with a peak on one and only one. There is a perfect correlation no matter what tool you use. But what we see is not the case, that these values are not the same. They don't correlate with each other. We have different GMB estimates depending on the tool we use. And you can tell me, of course, this is not, this is just values that in the end, they're different, they're shifted, but they will have the same information. We built machine learning models to predict brain age based on that. And what we found is quite interesting. For example, if you use ANTS with the general template, these are different. Each color is a different learning algorithm that are usually quite good at predicting brain age. And what we see is that on the mean, it's around 7 years. And remember that I showed you that the mean was around plus minus 5. And what we see if we use fMRI prep with FSL, we see 5.83. So there is a variability in how the models perform based on which decisions we took early on in the processing of the data. And at this point, I don't expect everyone to understand everything that is going on, because what happens here, and this is a slide I would like to show all the time, is that there are a set of basic skills that we need to handle in order to do these kind of projects. And it's quite hard to actually learn all of them deeply. So what happens is that, like we use, like I was saying, Spanish, que mucho abarca, poco aprieta. It means that if we try to hold too much, we're not going to go squeeze, which means that it's very difficult to actually learn how to use something in detail if we need to learn many things. We don't have time. If you start your PhD and within 3, 4, 5 years, you need to manage all of these things, it's quite hard. And you're not going to have time. You need to know about the data set, you know how to program, but you also need to know how to handle data, how to process the specific data that you're working with, like neuroimaging data with fMRI prep, FSL, SPM. And since we're dealing with thousands of subjects, usually we need to use computational clusters, which gives another layer of complexity. Then handling the data is not that simple. And of course, we need to learn how to do statistics, visualize, and finally write everything and put them in a paper. This is quite hard. So my goal for the rest of the talk is to show how to first plan the project, knowing that we're going to do ML, because this is where all the problem starts. And then how to extract features from neuroimaging data. And this is one of the toolboxes that we have. And then how to build models and estimate their performance. Okay, so let's move on with that. In order to plan the machine learning project, knowing that we're going to do machine learning, we need to understand how we're going to evaluate how our machine learning model works, because this is the essence of building machine learning models. It's like showing that it will work on unseen data. So what we do is this estimation of the performance that I shared before is called cross-validation. This is a specific type of cross-validation. It's called k-fold. The idea that we have our data with the features and the target, and then we do k-splits. In this case, k equals to 3, so we have 3 splits, and then we use 2 for training, we train the data, we pre-process, we train and we learn, we have our machine learning model, and then we take this last fold that we never touch, we use it as a test data, and then we have our error. And then we repeat, but instead of leaving number 3 out, we leave number 2 and number 1. So we have 3 error estimates, and this is our estimation of the error. This is quite good, because what happens if we only do it once, we might be very lucky in the way we partition the data. So we want to have several estimates, but not too many. Usually nice values for k-fold is 5 or 10, and that's it. So once that we understand that this is what we're going to do, we need to see what are the things that can go wrong and things that we cannot do. Because cross-validation provides an estimation of the generalization performance. It's like how our model will work on our unseen data. And usually this is overestimated. So basically, we're saying that our model, we estimate our model to work better than that is actually working. And this leads to what we call model failure, because if you promised or you tell me that you're going to work with an 80% accuracy, or you're going to miss by 5 years, and then I try it on my data and it missed by 10 years, I'm going to say, okay, your model is failed. And the biggest issue that we have is called leakage. And leakage happens when there is an information that from the testing data set, any sort of information that is known to the model at training time. So this can happen because of design issues, because we design our analysis incorrectly. That's one of the things that we see the most. The other one are coding errors. They exist too, but they're difficult to spot because you need to go through the code and see where, maybe a variable that shouldn't be there is there. So the machine learning model knows about the answer before even being tested. And the other is mostly human induced because we repeat the analysis too many times. So at this point is the user, the developer, us, the researcher, we know exactly what the testing data has. So we end up building a model just to satisfy that testing data and not any test data. So let's go a bit through all the things that can go wrong in this process. So I hope to get your attention why we need to design things properly and what things needs to be checked. The first one is quite simple. It's called test to train leakage. And it's the version number 1, which is basically sending the testing data as training data. So the model already saw what we're going to ask. So imagine that you take the exam and you already know which are the questions that you need to be answering. And you have the answers and the questions in front of you. You just memorize that, and then you go to the exam, you repeat. Of course, you pass, but you learned nothing about it. This is quite blunt. Usually this happens because of coding errors. But there is another version of test to train leakage in which we've seen it a lot. I just recently saw a paper published that actually does that. Let's say that we need to transform our data. So for example, we need to normalize our brains to a space, and we don't want to use any M&S space. We want to use some sort of like average brain of our population. So what happens is that we are transforming all the data at once, including the testing data. And if this data is coming from the future, there is no way that we could have transformed it and included it in our average. So this happens when we take our data out of the scanner and we do something that manipulates the data as a whole, and then we put it into our splitting process to estimate the performance. This is quite common in neuroimaging, for example, doing PCA or some sort of dimensionality reduction at this step because we have way too many variables. Even for the target, this is called leakage, and it will give another estimated model performance. Sorry. The next one is called test-to-test leakage, and this is similar to the previous, but in the end, what happens is that these samples that are here that are going to be tested, they are treated as a group, and they're also processed as a group. And the idea of machine learning models is that we should be able to predict all the samples independently. For implementation reasons, we just put all the data together and we ask for all the predictions at once. But if we do it individually or all at once, they should throw the same result. And this is where it starts to become a bit more complicated. I don't expect everyone to understand. I just want to scare you a bit about all the things that can go wrong so you do it properly from the first time. This is called test-to-test leakage, and basically is like averaging predictions from different models. So instead of splitting the data and then computing the performance, we just average all the predictions together and then they compute the performance only once. Many people do this with correlations. So they compute the values of a prediction, for example, brain age, for different samples in different folds, and then they put all the predictions and then they compute one correlation. What happens here is that we're actually summarizing evaluating several models at once. It's a meta model that is called a voting or ensemble models. And what we're doing is giving the training error of a model that uses several models at once. And it's not the testing error. And the last one that I want to show is called feature-to-target leakage. It's in a way when we define the target based on a feature. So let's say that we want to build a classifier to distinguish between old and young people. Okay, let's call it a bit like experienced and unexperienced people, and we put the threshold at 40 years old. Don't worry, I'm getting into the experienced people in a couple of months. So we have our age, which is the red bar here, and our target will be experience or unexperienced based on whether the age is 40. But then we make a mistake, and this age gets in the data, and it continues in the training, and it continues in the testing. The models are very powerful, but they're also very lazy. They're going to take the shortcut. So basically, it's going to use this value to continue to do the prediction. It's going to look at the age. So it doesn't matter whether we put the brain information or whatever information we put, the model is going to take the shortcut, and because of an implementation of mistake, there is a feature that is linked to the target that should not be there. So these are all sorts of mistakes that can happen because of the way we treat the data. And this is the key part. We are estimating how well a machine learning model works. We want to do it in a way that there is no leakage. So let's go back to the implementation of the project and what do we need to do. If you think, there are two steps. I mentioned the last one, which is the machine learning. But let's start with where do we get the information in a way that there is no leakage there. That is extracting features from the data individually for each one of the subjects in a way that they don't merge. So for that, we developed this tool, what is called Junifer, which is a feature extractor. And for the machine learning part, we developed another one. I will show it in a couple of slides. The first one is the Jülich Neuroimaging feature extractor, though we're going to extend it for more than just neuroimaging, but for the moment it's for that. It's not a pre-processing denoising tool. We have fMRI prep for that it will take all the information from the subjects and put it in a way that we can use it and compare it with each other. It's just a pipeline configuration tool. The pipeline is fixed, the steps are configured, and this is how the pipeline looks. I will explain all these steps one by one, but they're quite simple. The good thing is that it integrates with high-performance computing or high-throughput computers, so we don't need to learn how to use the cluster. We just need to tell Junifer the information about the cluster. We use state-of-the-art tools, so we're not reinventing the wheel. We're using whatever the community is using already. We just call it. We call it, we use it, and we wrap it back, so we don't need to take care of where the files are, how it works, what are the parameters. And of course, it's easy to use, because it's aimed for researchers with diverse background. We don't want you to learn yet another tool, another more complicated things. We want you to access all of the power that is already there in an easy way. And it's a no-code tool. Basically, we don't write Python code, we don't write MATLAB code, we just need to configure the pipeline using a very nice syntax I'm going to show now. I'm going to go quite fast now. Don't worry, the idea is that you have some of the information for you to go check the documentation work with it. The first one is the data grabber, in this pipeline, which is the interface between what is the data set and what we call the data object, which is the thing that we're going to work. It is the only component that requires understanding what the data set has. So if somebody else or a colleague or even us, because we have some data set that we already included, we don't need to understand how things work. We just need to I want to use this data set. So we work with patterns because all the file names, they usually follow a pattern, and what changes across pattern and this wildcard, this replacement, we can be the subject name, the subject ID, the session ID, or the task ID. And if you work with OpenNeuro, it can be accessed through DataLab. DataLab is a tool to access all this data, and the syntax is quite simple. This, I leave it here for you to work if you want to try it later. This is for the IOMIC data set, and what we do here is we define, I mean, this is the URI or the link to access this data set from OpenNeuro, and we define the patterns to access the files. This is done manually on purpose, but this data set we haven't included. So basically what we see here, we have the wildcard that is named subject, and we have also the replacement here. It said, like, we instruct Junifer to replace the keyword subject with whatever we're going to see later that is the subject. And we have here the BBMGM, which is the gray matter probability map. Then the next one is the data reader, which is a step that transforms parts that actually reads the file. But we don't even need to mention it. We don't even need to specify it, but you will see if you run it that at some point you will see some login output showing reading the data. And if it fails there, it means that there is a problem with the file. There is a pre-processing step, which call it pre-processing, but it's usually a data transformation step. It's optional. If you want to know an example of something that happens in pre-processing is with the bold data, we want to remove motion regressors, for example. Or we do some temporal or spatial filtering, or we want to compute the mask. We also have another way of transforming the data, that is to transform into a different MNI or even native space where we have the individual transforms. And this is how we specify. This is quite complex in the pipeline, but this is how we say that we want to detrend, standardize, and remove all the motion regressors, or all the white matter and CSF regressors that were computed with fMRI prep. This is a low pass and high pass filtering, and this is because this is to compute the mask using the subject's gray matter probability map with a threshold of 0.2. That means that everything under 0.2 will not be considered for the computation of the markers. And the next is to define the markers. We have quite several markers now. I think this slide is a bit old. We have more, and we keep adding things to the list. And it's quite simple. We just need to define the marker section, and we put all the markers we want. This is a name that we're going to use later to distinguish which markers. We can put whatever we want. This is a parcel aggregation. So basically apply a parcellation and perform an aggregation function. We define the parcellation. There is a list of parcellations that are embedded in Junifer. And this is because we want to apply the mean. But the thing is, like, since we already did all the pre-processing, reading, and all the stuff of the data, why just only one? If we need to compute more, we can compute. What we're doing here is computing the standard deviation across the boxes in the ROI instead of the mean. And then we can do the same thing with another parcellation. This is the Shen 2019, and this is the Shen 2013. Let's say we want to compare to see which is the best parcellation that we can use for our problem. And that's it. Once we define all the markers, we need to define where do we store it. And once we store it, we have this. This is the one to be used. It's quite fast. We have this one, which was a bit slower. It's an older version. So it's quite simple. We define the kind of storage, and then where do we want our files to be stored. This is in my computer. This is. And in the end, we put everything together. I removed the fMRI prep because this is part of a BBMGM. This is part of the example I'm going to do later for a prediction. And in the end, we have this file. It's a text file. This is called YAML. And now just run. So we execute this command, Junifer run. And if you see, this is the YAML file. And here we'll see the element. The element to be run is what we are actually going to replace. So we have one replacement that it was subject. So this is the subject we're going to run. And this is just to run a test. What's happening is it's going to take the subject and replace it here. So this is the file that is going to be used. And this works. And what happens? They said it's going to be easy. Now I need to go for all the subjects. No. If you just don't put the element, it will run for all the datasets. So it will go for the 900 subjects or 1,000 subjects that we have in Neuriomic, and then extract the same feature, create an individual file for each one of the subjects. But if we then run the collect, it will just take all of the individual subjects and put it into one file, so we don't need to even go and look for many files in the file system. And since we're dealing with 1,000 subjects and these things might take time, we might want to parallelize this. So we have the queue. We can define a queue section in which we actually tell to use one of the computational clusters that we have in Jülich, which runs on HT Condor, or we can even use GNU Parallel. We have plans to support others, but since we don't have use cases so far, we haven't really worked on it. And this is quite a new library that we have. So if we do Junifer queue, this will just queue in the cluster, run, and at the end, we will have all the results. So that's it. This is easy. We just need to define which markers we want, and we will have one file with all the information. So let's go back to how to do the whole project. We already covered this part. We got our features. We can define many markers. Let's see how we can do the machine learning part with Julearn. Julearn is a user-oriented machine learning library. The idea is to do machine learning with less amount of code than scikit-learn. This is not a replacement from scikit-learn. It works with scikit-learn. If you don't know what scikit-learn is, it's one of the most powerful libraries that everyone is using to build classical machine learning models. It's Python-based, but it requires quite some coding. It's very flexible. They focus on the machine learning. What we're doing is adding a layer on top of the complexity that scikit-learn has to make it easier to access scikit-learn. The idea is to build the same pipelines easily, avoiding data leakage, because Julearn will not allow you to cheat on that aspect or to make mistakes, because everything will be quite checked and protected from that. That doesn't mean it's leakage proof, because you may still find ways to hack it, and it might be that there are some things that we haven't considered yet, but it's quite safe in that sense. And the extra functionality that we have is that we can do confound removal. If you read any paper that is using this, now it's easy. We have data typing, so instead of putting columns 1 to 10, we can say these columns, they are a type because they come from the structural brain imaging. These columns come from the functional brain imaging, so then we can transform them independently. We can also inspect the models to see what's going on, why it works, why it doesn't work, and then we have tools to do model comparison. And so, like the name here, it says, this is what our group leader defined. You need to keep calm and just run cross-validation, which is basically doing this. So let's look at a simple binary classification example to see how this can be done, and then I will show you how we can do this with neuroimaging. So remember, we have our X, our Y. We want to find an association between the X and the Y, basically predict Y given X. And the first example I'm going to use, flowers. This it's just some Python code. If you are not very familiar with Python, this is just importing, telling that we want to use these functions. And what I'm doing here is loading this dataset iris, which is a dataset of flowers with species, that there are 3 species here, and just filtering so we only have 2 and we have a binary classification. The next step, if we're using Julearn, it's just quite simple. We define which is our X, so the names of the columns that represent the predictors, the features, and our Y, which is the column that represents our target, what we want to predict. This is the CV. This is from scikit-learn, is how we define the cross-validation strategy. We're using a stratified k-fold with 5 splits. That means that we partition our data in 5, but we try to keep the ratio of samples in each class equal across the splits. And then we just define our model using our simple pipeline creator. We tell it's a classification, so we have two classes. Keep in mind that if we look at the data here, we're going to see this in the data. It's not going to be 0, 1. It's going to be strings, labels. Then we do a Z-scoring, and we say we want to try a support vector machine with this kernel linear and these parameters. I will explain a bit about this later. And then we just run cross-validation. We call the function. We ask for the scores, the model, and the inspector. By adding here, we say this is our X, this is our Y, this is our data, and this is our model. And that's it. We can take this. This is going to be a data frame. And then we can look at the values there. We have all the test errors. And we can do, for example, an average or a nice plot and show that. So this is how simple it is to actually evaluate if this learning algorithm is good for distinguishing between these two species based on these three variables. That's it. Now, the main issue is like here it's a binary classification, and I'm assuming that we have like 50-50 chance of selecting randomly the right class. There are some other problems that are a bit more complicated. And the other question is like, I just did here, I said an SVM with a linear kernel and these parameters here. But is this the best model? This is the first model I try. If this model is not 100% accurate, but it's like 80% accurate, this is the best I can do. So actually building a machine learning model is not just trying one, but it's trying several and picking the best. If you try too much, of course, we're going to overestimate. But let's see how we can do this easily with Julearn, how we can build several models and compare them. This is one of the key functionalities that we have. So basically, I have here three creators. One is a support vector machine, no parameters. The other one is a random forest, no parameters. And the other one is a support vector machine with a linear kernel. And then it's just running the same thing. We run cross-validation of the three. Here it's the creator 3, creator 2, creator 1. And to the scores, I just put a name so I can have it when I plot it, because then we can call this visualization function where it's plot scores. It will show an interactive. Here it's not, it's just a screenshot. And we will see the dots with the distributions for each one of the models across the evaluation folds. In this case, I tried 25 folds because it's 5 times 5-fold. I repeat this 5 times with different random seeds. And what I show is the balance accuracy. So what we see in this plot is that the support vector machine works with an average around 93, 92. The random forest, it's around 89. And the support vector machine with a linear kernel is a bit better, around 94. But if you encounter any reviewer, they will ask, okay, but what about p-values? Can you do a t-test? Can you do anything? And it's not that simple because machine learning models have the problem that the samples are used sometimes as training, sometimes as testing, so the independent assumptions are not met. So what we have is when you click on this button, you will have the statistics here, and this is a p-value that is corrected for machine learning models. So this is not a t-test, this is a specific test, and we have the reference in the documentation. And this is the corrected p-value, basically doing Bonferroni over this p-value for the multiple comparison test. So you can take this and just report these p-values that you have here and say, okay, this model, it's not significantly different from the others. It looks better, but it's not significant. Now, if we add another model that is worse, this is quite an easy problem. We can say which one is the best working model. Now, I talk about the support vector machine and the support vector machine with the linear kernel, and this is because we have what we call hyperparameters. Hyperparameters are, they call hyperparameters because of the parameters of the model, but they are to control the aspects of the learning algorithm. So for example, in a support vector machine, we have the kernel type and C. These are two parameters. The random forests have like the number of trees and the depth of each tree and many more. So these hyperparameters can prevent what we call overfitting, which is a complete memorization of the training data. If we memorize the training data, the rules are going to be specific, but then when we have testing data, they're not going to be work. And they must be tuned. That means if we try one model without changing the hyperparameter, how do we know that this is actually the best configuration for this learning algorithm? So we need to tune them, and this is done through what is called nested cross-validation. So basically, we have our cross-validation to evaluate the performance, and we just take our training data and we do the exact same process to select the best hyperparameter. And the thing looks a bit like that. If you want to implement it yourself, you need to implement nested for loops, and it becomes a bit complicated. And of course, remember, one mistake, you get leakage, and then you overestimate the whole thing. So how do we do with Julearn? Well, in particular, you don't need to do much. You just need to give a list of hyperparameters to be tested. Here we have several options for C. This controls the linear kernel of the SVM. But when you have a kernel that is different from the linear, there is another hyperparameter called gamma. And you can also try several options. And since you want to also decide between these two, which one is the best combination, you can also have a list of pipeline creators to the model, and Julearn will do the nested cross-validation with these two hyperparameter sets and find which one is the best and give you the scores that you can actually go and present. So, yes, great. But I want to hear about neuroimaging. I don't want to hear about flowers. I don't care about flowers. Okay, let's move on. It's actually simpler than I expected. How to use Junifer in combination with Julearn? Let's say we want to predict sex, biological sex, from gray matter volume. It's actually a quite simple model. It works very well. And remember that I gave you the tools to compute the gray matter volume, and I gave you the tools to do machine learning. So let's combine everything together. The first thing is to use the Junifer storage in Python and read the data frame. This is the name that we put. It includes here the type of data at the beginning, but there are ways of finding this. And when we read this, we have a data frame. Indeed, we have a table in which we have the subjects. And here we have the 1,000 columns from the Schaefer 1000 parcellation. So we have our data frame with the features. Let's include the demographic variables. This comes from Aomic data from OpenEuro. Everything is there. The only thing that we're doing here is changing the participant ID to subject to match what we have in Junifer. And this is how it looks. And here we have the sex, among other variables, but we don't care about them because, as we know, in Julearn, you can just choose which variables to use. And then I'm going to read also I'm going to read the total intracranial volume because I have the slight impression that this has something to do with the prediction. So we read that, and then we select the X is the columns of DF, which is the variable here. So basically the 1,000 parcellation names. We put all the data together in the DF full data frame. We create our support vector machine. We tune the hyperparameters. We run the cross-validation, and we get the scores. And this is how the scores look like. It's just a data frame with many, many columns, and this one is the test score. This is what we're interested in. The train score is how well the model works on the training data. A train score of 1 means that the model can predict the training data very well. Now, we want the test score, which is the test data. And here we have the 5-fold that we tried. So we see that the model works quite well. Let's see why. And why do I include the total intracranial volume is because I have an impression that this has to do with the prediction. So one thing we can do with Julearn, and I told you that we have the inspector, we can take the prediction for each one of the folds. And then I get the predictions with the actual sex variable and the estimated total intracranial volume, and I put them all together. This is a bit of pandas data frame handling, but you can do with any other tools you have. These are all data frames. These are Excel spreadsheets. And then I make a plot in which I plot on the x-axis, I'm going to plot the sex, the hue, that means the color, whether they are correct or not correct, and the y I'm going on the y-axis, I'm going to put the total intracranial volume. And what we see here is that it's actually quite interesting. The corrected classified males that we know they have bigger heads on average, chest head, total intracranial volume, are usually have a higher intracranial volume than the incorrectly classified. But this reverses for the females. So the total intracranial volume of the incorrect is higher than the correct. So what we see here, this trend, what shows us is that overall, what the model is capturing is a proxy to the total intracranial volume rather than actually differences in the gray matter volume or the regional gray matter volume, which means that we might need to consider and correct for the total intracranial volume. And if you want to do that, it's a part of the documentation of Julearn. You can find how to do confound removal. So you can decide that the total intracranial volume is a confound and I want to remove it from the data before doing the prediction. And then you will see whether there is actually an association between the regional gray matter volume and the biological sex. So I'm going to stop it here. Takeaway message: plan your analysis even before touching the data. Check with someone that knows about ML and be careful. If you build a machine learning model or somebody built a machine learning model or 2 or 3, they know about ML, but being an expert in ML takes way more time, way more research, way more experience. If I learn how to drive my car, that doesn't mean that I can fix the engine of a truck. It takes another set of skills to find what is wrong and whether it's properly done. And there is a lot of misinformation going around. So please check with someone. I will give you some information we have. If you want to check with us, we're also available for that. Second message: after pre-processing, use Junifer to extract features. You'll make sure that you don't mix data around and you do everything in the same way, because it is easy to do ML incorrectly without knowing. And what happens is that usually leads to good results. So we're tempted to publish that because we found actually a model. We tend to say that if it's perfect classification, we discard it. If it's chance level classification, we discard it. But anything in between gets published. And the question is, how do we know that this is not overestimated or there is a mistake? And Julearn will help you prevent mistakes. So if you want to check the 2 toolboxes, we have the documentation there. The one from Junifer, it's there is quite some information there how to extend. It's more technical, but we are working on it. It's actively being developed. Julearn is a bit more mature library, and there is a lot of information on how to understand machine learning, how to build the models, how to do some of the things that I mentioned step by step, explaining the concepts. And if you really need to consult with someone, we provide this consultancy on machine learning every 2 Thursdays. Unfortunately, for some of you, this is at 2 p.m. Central European time. You might need to get up early, but you can come with questions. We don't, it's not a collaboration. We just try to answer and give information for free, meaning that we're not going to be co-authors. We're not going to do your analysis. We're just going to help you plan, execute, or check your research. And that's it. That's my email if you want to contact me. And thank you very much.